Hello and welcome back. So I'll be continuing my introduction to the immune system. That is a structural overview, and this will be part 1.2a, and we'll be talking about lymph nodes as secondary lymphoid organs. So as we discussed, the general structure of the immune system includes primary lymphoid organs, secondary lymphoid organs, and tertiary lymphoid organs. And today we'll be concentrating on the secondary lymphoid organs. Now, coming to the lymph nodes as secondary lymphoid organs, we'll talk about the general structural overview of lymph node. We'll talk about lymph node structure and try to understand how and why the structure is the way it is. We'll talk about the role of lymphoid tissue inducer and lymphoid tissue organizer cells in the formation of a lymph node. Then we'll talk about fibroblastic reticular cells or FRCs and their heterogeneity based on location. The chemokines they produce, particularly homeostatic chemokines. Understand how the compartments within a lymph node are populated based on the gradient of these homeostatic chemokines. Then we'll delve into how a germinal center is formed and how this can be understood as a coordinated T and B cell dance. Then we'll finally talk about the role of CXCL12 high fibroblastic reticular cells in the germinal center and how that affects germinal center functioning. And then finally we'll talk about some other quote-unquote other homeostatic chemokines. Now, the general lymph node structure is as follows. There are certain structural compartments within the lymph node. So these are the efferent lymphatics coming into the lymph node, as you can see with the arrows, and this is the efferent. So these are the efferent lymphatics coming into the lymph node, and this is the efferent lymphatic going away from the lymph node. So this would be the draining tissue here. Okay, and within this lymph node, as you can see, we've got sub structures. The outermost is called the cortex. Then we have the paracortex, and then we have the medulla. And this area where the efferent lymphatic is located and the draining or the blood vessel that supplies the lymph node is located is known as the hilum of the lymph node. So we have some spaces within the lymph node. The one is the subcapsular sinus, that is between the capsule of the lymph node. So this is the capsule of the lymph node and the cortex. Then we have the cortical sinus, which is located in between the different parts of the cortex. Then we have the medullary sinus located within the medulla of the lymph node. And then, as I just said, there's the hilum. Now, this is something that I've actually discussed in an approach to generalized lymphadenopathy as well. So we'll, in this screencast, I want to actually highlight the hows and exactly the whys of how this happens, how the structure comes to be. So these are high endothelial vessels liquidated within the lymph node, and this is how WBCs from the blood enter into the lymph node and exit the lymph node back into blood. So we need to understand exactly how this happens in order to be able to understand the structure better and to not have to memorize it. Also, we'll see this plays a very crucial role in understanding antigen dependent T and B cell development later in the series. So how does the lymph node form? So it is through an interaction between two types of cells. We have a lymphocyte precursor derived cell called a lymphoid tissue inducer or an LTi cell, and we have an embryonic FAP positive mesenchymal cell derived cell which is called a lymphoid tissue organizer cell or an LTo cell. So an LTi cell produces a particular cytokine called lymphotoxin alpha and beta. This is a dimer, but I've shown it as a single protein here that acts on its specific receptor on the lymphoid tissue organizer cell, and this causes the embryonic FAP positive mesenchymal cells to differentiate into your lymphoid tissue organizer cells, and these lymphoid tissue organizer cells then convert into the various reticular cells that are located within the lymph node. So RC stands for reticular cells, and these populate different compartments of the lymph node and start producing specific chemokines. These chemokines are as follows: CXCL13, CXCL19 and 21, CCL19 and 21, CXCL12. So we need to understand exactly how this results in the lymph node that I described in the beginning of this screencast. This is a lymph node as it is forming. So the lymphoid tissue organizer cells have converted into reticular cells or fibroblastic reticular cells, and they have formed a meshwork within the putative lymph node that is to be formed. Now, within this meshwork, the fibroblastic reticular cells are heterogeneous, and this heterogeneity is actually a function of their location. Where are they located within the putative lymph node is what determines what type of chemokines they produce. Okay. So going forward becomes very important to know what are chemokines, and chemokines basically are a family of proteins that are all very small, that is 8 to 10 kilodalton in size, and all contain cysteine in their primary structure. So the cysteine residues are located at specific locations within each chemokine, and thus they determine the fine type of folding that occurs within that particular protein and the eventual 3D structure that characterizes that protein, that chemokine. This is because cysteines are able to form intra protein cysteine adducts. Now, this divides chemokines into 4 basic subfamilies based on the number of cysteine residues and their relative location within the primary structure of the chemokine. These are CXC, CC, CX3C, and C families. This basically, the X stands for an amino acid other than cysteine. So CXC would be 2 cysteine residues separated by one single non-cysteine amino acid. Similarly, CC would mean that 2 cysteine residues are right next to each other. CX3C would mean that there are 3 non-cysteine amino acids between 2 cysteine residues, and C means that there is a cysteine that is separated from other cysteine residues by a large number of intervening amino acids. Now, the chemokines bind to specific receptors, and these receptors naturally are called chemokine receptors, and these are transmembrane receptors. Specifically, they are G protein coupled receptors. Therefore, any cell that expresses a chemokine receptor moves along the gradient of chemokines that are being produced from a particular source. So it moves towards increasing concentrations of that particular chemokine. Now, having said this, chemokine receptor interactions are highly specific, and therefore it is important to know them in order to understand how immune cells are located in relation to one another. So there are three basic chemokine interactions that we need to understand before we go forward and understand the structure of a lymph node. So the first is CXCL12, the receptor for which is CXCR4. Then we have CXL13, which is the receptor for which is CXCR5. Then we have CCL19 and CCL21, both of which bind to the common receptor CCR7. Then we have CCL20, the receptor for which is CCR6. So it's actually very easy to remember. If you know CCL12 and binds to CXCR4, you add 1, it becomes CXCL13, and you add 1 here, it becomes CXCR5. So that becomes very easy. Similarly, if you know that CCL21 and 19 bind to CCR7, then you subtract 1 from 21, you get CCL20, and 1 from CCR7, you get CCR6. So the receptor naming is based on the sequence in which they were discovered. So it is arbitrary, and it needs you need to remember it. Now, these chemokines are constitutively produced by tissues in absence of any immune activation, and therefore they are called homeostatic chemokines. CCL20 and its receptor CCR6 are important in mucosal immune responses. Therefore, I'll be talking about them at a later date. So we'll for now restrict ourselves to the remaining 4 chemokines and their respective receptors. There are some other quote-unquote homeostatic chemokines. These are basically CCL14, CCL25, and CCL27. They play minor roles in most circumstances, and therefore I'll just highlight their roles at the end of this screencast. Fibroblastic reticular cells are also called reticular cells and differentiate into multiple subtypes based on their location. So what we'll do is we'll talk about certain fibroblastic reticular cells based on their location. So the ones that I want to highlight initially are marginal FRCs, T cell zone FRCs, and medullary reticular FRCs. So we'll talk about the specific production of CXCL13, CCL19 and 21, and CXCL12, and you'll understand then what role these FRCs play, and we'll understand their function. So marginal FRCs are located in the subcapsular sinus and are characterized by the expression of BP3 and a protein called MADCAM. Okay, BP3 is also called FGF3 binding protein, and the T cell zone FRCs are characterized by BP3 expression but not by MADCAM expression, and therefore are located more towards the medulla but not exactly in the medulla. Cells that are leptin receptor high but do not express BP3 or MADCAM end up in the medullary, exactly in the medullary region of the putative lymph node, and that they are then called medullary reticular cells or medullary fibroblastic reticular cells. So marginal cells express CXCL13. They attract CXCR5 positive cells. This includes Th2 T cell response corresponding dendritic cells, and we'll see how this is relevant when we talk about lymph node pathologies and how to interpret the histopath of lymph nodes. They do not express CCL19 and 21 or CXCL12, so they are going to attract only CXCR5 expressing cells. So that is to be expected. So now T cell zone FRCs express CCL19 and 21, and they express CXCL12, but they do not express your CXCL13. So naturally, what will end up happening is all cells that express CXCR5 are going to get attracted towards the marginal FRCs in the subcapsular sinus, and all cells that express your CXCR4 and your CCR7 are going to be attracted towards the middle. So, and finally, the medullary reticular cells only express CXCL12. So any cells that express only CXCR4 will get attracted towards the medulla, and actually they leave the lymph node to the efferent lymphatics. The marginal zone fibroblastic reticular cells express IL-7, which supports a specific type of innate lymphocyte cell known as ILC3 at this location, and they produce RANKL, which supports sinusoidal macrophages. So if asked what are the cells that are located within a normal subcapsular sinus within the lymph node, it includes ILC3 cells, it includes sinusoidal macrophages, and if the local tissue has a Th2 polarized response, it can include dendritic cells that are produced during a type 2 immune response. Similarly, T cell zone FRCs secrete IL-7, they secrete IL-15 and IL-33. This is during homeostasis. This supports 3 cell types here: lymphoid tissue inducer cells, which we've seen are what started this process of, other ILCs, particularly ILC1 and ILC2, and dendritic cells, which are resident within the particularly plasmacytoid dendritic cells, which are resident within the T cell zones of the lymphoid and final lymph nodes. Okay. Now there is another type of FRC here called the TB FRC or TB junction FRC, which is BP3 positive but BATCAM negative, just like a T-zone FRC, and it has an expression profile that is very similar to a T-zone FRC. It expresses APRIL and IL-6 and supports plasmablasts, and we'll see this actually is a niche for supporting plasmablasts after germinal cell development, and that is where it becomes relevant. Now, thus the compartments within the lymph node get populated by specific cells. So the subcapsular sinus, which contains your marginal FRCs, is going to express high concentrations of CXCL30 because marginal zone FRCs express CXCL30. So naturally, any cell entering into the lymph node expressing CXCR5 is going to get attracted towards the capsule of the lymph node or the subcapsular sinus. The influx of lymphatic fluid causes a mechanical force, that is a pressure, since this is a fluid medium. That is what actually results in a separation of these cells from the subcapsular sinus and this potential space containing fluid, which is called the subcapsular sinus. Thus, B cells, which are the cells which express CXCR5, get sandwiched between the this fluid here and your T cell zone. So B cells primarily are located within the cortex of the lymph node, and that is what we call the cortex of the lymph node. Now, this forms the B cell zone or the cortex of the lymph node. Similarly, since the medulla of the lymph node is, or the medulla and the paracortex is populated by T zone FRCs, and T zone FRCs express CCL19 and 21, any cell that is expressing CCR5 is going to get attracted towards the T cell zone. So as the name would suggest, it is obviously T cells that express CCR7 normally in homeostatic conditions, and therefore are attracted towards the T cell zone. So as a result, you end up having a division here, this being the cortex, which is the B cell zone, and this being the paracortex, which is the T cell zone. So this is the reason why we have the cortex and the paracortex within a lymph node, and the cells that populate the locations wherein. Now, the medullary sinus contains medullary FRCs, also known as simply as medullary reticular cells, and they, as I've just said, express only CXCL12. So any cell that expresses CXCR4 is going to get attracted towards the medullary sinus and then move through efferent lymphatics outside of the lymph nodes. Okay. Now, as I've just highlighted, there are also some round structures located between the B cell zone. These are actually germinal centers, and they seem to have different structures, as you can see. So we need to understand what exactly is happening here, and why does the structure look like this. Okay. So we'll just fill in the blanks that we've left. So the germinal center is actually populated by 2 specific types of follicular reticular cells. They are then called follicular dendritic cells. So they become FDCs rather than FRCs. So one is known as the dark zone FDC, and then we have the light zone follicular dendritic cell. Both of them have a phenotype similar to your TB cell FRC, as TB junction FRC, as well as the T zone FRC. Now, dark zone FRCs express only CXCL12. So naturally will attract CXCR4 expressing cells, and light zone FDCs express only CXCL13, so they're going to attract CXCR5 expressing cells. So naturally, any cells that land up in the dark zone are going to be CXCR4 receptor positive, and any cells that land up within the light zone are going to be CXCR5 receptor positive. Okay, so let's see what the function is. Okay, so as I've just discussed, T and B cell junction FRCs express APRIL and IL-6, which supports plasmablasts that are produced within the germinal center, and we're just going to talk about what a germinal center is. Okay, so a well-coordinated chemokine dance is orchestrated between B cells and T cells, and that results in the formation of a mature germinal center. So on this side we have our subcapsular sinus, and on this side we have the medullary sinus. Naturally, the subcapsular sinus is going to contain our marginal FRCs, and this is going to contain the medullary sinus is going to contain our medullary FRCs. So under normal circumstances, as I've seen, because the subcapsular sinus contains your marginal FRCs and they express CXCL13, your B cells, which are CXCR5 positive, are attracted towards it and keep sitting there. Now, medullary sinus and the paracortex contains your T-zone FRCs and medullary FRCs, so they express CCL19 and 21 and attract T cells which express CCR7. So that is why this is the initial orientation that we have. Now, once antigen exposure happens to a B cell, so once an antigen the B cell interacts with an antigen, it moves. So it changes its expression profile and it moves. So naturally, in order to move to the junction between the cortex and the paracortex, it has to increase expression of CCR7, because obviously we have CCL19 and 21 being high towards the medullary or the paracortex area. Now, that same antigen coming in from the afferent lymphatics is also processed by dendritic cells, are located within the paracortex and shown to T cells, and the details of which will come to later. That causes your T cell, which was initially just CCR7 high, to shift towards the junction between the cortex and the paracortex by increasing the expression of CXCR5 and decreasing the expression of CCR7. Now this B cell and T cell are right next to each other at the cortical and paracortex cortical junction, so this would be called the TB junction. Okay, now if the T cell and B cell have been activated by the same antigen, the B cell is able to further give signals to the T cell, which results in further increase in expression of CXCR5, naturally causing the T cell to move towards the cortex, and it also induces the expression of your sphingosine 1 receptor type 2, which again causes the T cell to enter into the cortex and sit there. Now the T cell within the cortex starts to express CCL 19 and 21. So now there are two sources of CCL 19 and 21. One is your T zone FRCs, which are producing CCL 19 and 21 in this gradient, and you have T cells within the cortex which are producing CCL 19 and 21 within the cortex. So now what ends up happening is that we now have an additional smaller gradient of CCL 19 and 21 in the cortex, and any cells, for example this B cell that has interacted with an antigen and then with a T cell, as if it is expressing increased CCR7, is going to get attracted towards this T cell within the cortex. So that is what ends up happening. The B cell increases its CCR7 expression and sphingosine 1 phosphate receptor 2 expression, resulting in T cells and B cells co-localizing within the cortex, and this through subsequent events ends up forming the germinal center. Now a final point here is that after this T cell B cell interaction within the TB junction, initially there is a transient increase in CXCR5 expression by B cells, but once antigen processing is complete, that they shift towards CCR7 and sphingosine 1 receptor 2 positivity. So those are the final details and they can cause confusion, so you can ignore this for now. Try to understand what I just said previously. So this ends up forming the beginnings of a germinal center, or this is the primordial germinal center. Now a cortical aggregate of small lymphocytes without a germinal center within it is called a primary follicle. Once a germinal center develops, we call it a secondary follicle. So initially this is the primary follicle, and once it starts developing further subdivisions, that is the dark zone and the light zone, it becomes a secondary follicle. Now this is enabled through specific interactions, and we'll just discuss those interactions. So a CRC is a CXCL12 high reticular cell. So naturally this expresses CXCL12, because that is what the name suggests, which is now a source of CXCL12 within the B cell zone. CXCL12 was normally being produced by the FRCs located within the medulla as well as the T cell zone only. Now what ends up happening through the subsequent steps is B cells convert into a type of another type of cell called a centroblast. Now centroblast is, as the name would suggest, has a morphology of a blast and therefore has a high nuclear to cytoplasmic ratio, as is seen in the diagram. So it's mostly nucleus. Because of that, when seen under the microscope, it looks dark because the nucleus stains it's intensely basophilic. Now T cells and B cells interact with each other, and these T cells convert into a specific type of T cell called T follicular helper cell, and it is these T follicular helper cells that help centroblasts go further ahead and convert into centrocytes. So now centrocytes, as the name would suggest, have more cytoplasm, and therefore their NC ratio is more towards the cytoplasmic, is lower, therefore they appear lighter. Okay, now some of these centrocytes convert into plasmablasts, and as we've seen before, after this these plasmablasts are going to go towards the TB junction. Okay, now this is your secondary follicle, and as you can naturally imagine, areas that are rich with B cells, T cells and centroblasts end up forming the dark zone. So this is your dark zone, and areas that contain your centrocytes and plasmablasts end up becoming the light zone. Okay, the light zone is further divided into two parts, your apical light zone and the basal light zone. That is a discussion for specific discussion under, we'll talk about it under antigen dependent B cell activation. Now, some centrocytes undergo apoptosis, whereas certain centrocytes are selected, and apoptosis happens within the basal light zone. Okay. Now, centroblasts and your T follicular helper cells are CXCR4 high, whereas centrocytes and plasmablasts are not. Okay. So this is exactly how a lymph node forms, and this is what a germinal center looks like, and this is why it has dark zone and light zones. So we've talked about the general structure of a lymph node. We've understood the hows and whys of how the lymph node structure is the way it is. We've talked about the role of lymphoid tissue inducer and lymphoid tissue organizer cells in the formation of a lymph node. We talked about fibroblastic reticular cells and their various subtypes and their heterogeneity based on location, the chemokines they produce, specifically the homeostatic chemokines. We've understood how the compartments within a lymph node are populated based on the gradient of these homeostatic chemokines, not cytokines. Okay, so we talked about the general structure of a lymph node. We talked about and we understood the hows and whys of how that structure develops. We talked about the role of lymphoid tissue inducer and lymphoid tissue organizer cells, the fibroblastic reticular cells that arise from these lymphoid tissue organizer cells, the heterogeneity of the subtypes of the FRCs based on their location. We talked about chemokine production, the specifically homeostatic chemokines, and we've understood how the compartments within a lymph node are populated based on gradients of these homeostatic chemokines. We've talked about how a germinal center develops briefly. We talked about the role of CXCL12 high FRCs in how in the functioning of a germinal center, and how that results in a basal dark zone and apical, or rather a dark zone and a basal light zone and an apical light zone. we'll get in more into this later. And now we're going to talk about other homeostatic chemokines. So there are three other homeostatic chemokines that we need to talk about. Now, unlike the chemokines I just talked about, these have specific locations during innate immune activation or inflammation. So these include CCL14, which is diffusely expressed, attracted to CC receptor 1 because it shares 74% of its sequence with two other CCA chemokines that we'll talk about later, that is CCL3 and 4. Then they are expressed primarily, they attract primarily macrophages, but they don't function as chemokines. They actually function as a macrophage growth factor. So technically, it's not really a chemokine. Then there is CCL25, which is produced by thymic epithelial cells within the thymus and small intestinal epithelial cells. Most prominently, it is produced within the duodenum, and then the expression tapers off as you go into the jejunum and is least within the ileum. The corresponding receptor is CCR9. The receptor is expressed by thymic T cells, which causes T cells within that allows for T cell development within the thymus, but this is a minor role. That is why we've not discussed it in our thymus video. Then it is also expressed by dendritic cells and macrophages. This causes homing of T cells, dendritic cells, and macrophages to the small intestine in specific situations. It is also expressed only by activated B cells, and that expression of by activated B cells causes the influx of activated B cells into inflamed tissue, allowing for local antibody production. CCL27 is expressed by keratinocytes, basal epidermal cells within the skin, and thymic epithelial cells. The corresponding receptor is CCR10. They are primarily expressed by memory T cells and allow homing of memory T cells to the skin. Okay. So these are the other three homeostatic chemokines. This is just for completion's sake, unrelated otherwise to the functioning and structure of a lymph node as a secondary lymphoid. So that brings me to the end of this screencast. Thank you for watching. And next time we'll discuss about other cell populations within the lymph node.